Thank you for the introduction. And hi everyone, my name is Dongyun Kim from Seoul National University in Sungyeon Kim's lab. And it's my great honor to have an opportunity to have a presentation of my works, our works for amore퍼시픽 Great Global Next Generation Research Award lecture. And I'm also so grateful to the committee for giving me the chance today. The title of my presentation is Neural Circuit Mechanism for Mechanosensory Feedback Control of Ingestion. So basically it is the works in neuroscience field, and the topic is control of ingestion, which is, very important but difficult in our everyday life. So the question is, how do we stop eating when we are full? So in other words, how do we stop eating after eating? So here's the background. After we eat, the food travels our body, which especially the digestive tract organs, which include the oral cavity, esophagus, stomach, and intestine, as you see here. And along the journey of the food, it generates multiple sensory signals like taste and nutrients, which are chemosensory signals, and also the texture or the distension of the internal organs, which are the mechanosensory signals. And these signals goes to the brain via the nervous system and computed in the brain, and finally let us to regulate our ingestive behaviors like eating and drinking. And we call this series of phenomena as negative feedback control of ingestion. And as neuroscientists, our question is, what is the underlying neuromechanism for this phenomena? And among these multiple sensory signals, today our work is focusing on the mechanosensory signals from the digestive tract distension. So actually, people have long known that gastric distension per se could suppress this appetite. In this remarkable paper, which was published 60 years ago in Science, Neil Miller have introduced a series of experiments from his lab, and where they used these small Rubbermaid balloon inside the stomach of rats, which we call gastric balloon. And then they trained the rats to press the lever to get the milk drop when they are hungry. So normally, the rats press the lever vigorously to get the food. However, interestingly, when they inflate the balloon, the number of the lever pressing was decreased, which means the suppression of the appetite of the rats by the gastric distension, the mechanosensory signals. And this phenomenon has been utilized to develop this gastric balloon therapy for treating obesity, which is approved by FDA in 2015. And in this therapy, this saline-filled balloon helps the people eat less and lose their weight by potentiating the gastric distension signals. However, which neurons in the brain receive the mechanosensory signals from the body and regulates the ingestive behaviors have not been identified yet. So firstly, we wanted to find the neurons that are activated by consumption. So by labeling the neuroactivity marker phosphoprotein, we found that specific neurons in the brain area called the parabrachial nucleus that especially express the gene called prodynorphin are specifically and potently activated by water intake. So from here, I'm going to call these neurons as PB-PDINE neurons, where PB stands for the parabrachial nucleus, the brain region, and the PB-PDINE is gene name. And then we we wanted to record the in vivo neuroactivity of the neurons by using a technique called fiber photometry, where we used the calcium indicator GCAM and collected the emission light from the neurons to measure the neuroactivity. And we found that these neurons not only activated by the water intake, but also activated by consumption of various types of liquids and solids, including liquid foods or even the silicone oil and the chopped food and high-fat diets too. And they tested whether the neurons receive the mechanosensory signals from the body. And we found that touching the tongue of the mice and insertion of the gavage needle through the esophagus, and also the distension of the stomach all activated the neurons, which means that these neurons monitor ingestion and receive the mechanosensory signals from the body. And the next question was how the individual PB-PDINE neurons represent these signals from different organs and ingestion signals. In order to record the activity of PBPD neurons at single cell level, we conducted in vivo two-photon calcium imaging, where we used the GCAM calcium indicator gain and imaged the individual PBPD neurons under the two-photon microscopy. And we found that most of the PBPD neurons are activated by water consumption, which is consistent with the first data, and also by the gastric distension and oral delivery of the small amount of liquids, which is constrained to the mouth. And what if we combine these three data? We found that the majority of the PBPD neurons, more than 80%, were activated by all of these stimuli, including water consumption and oral delivery and stomach distension, which means that these neurons represent the integrated sensory signals from the mouth and stomach to monitor the ingestion. And the next question was, what is the upstream neural pathway for mediating the mechanosensory signals? In order to figure out an upstream neural pathway, we conducted a rabies virus-mediated input mapping study, which enables us to visualize the upstream neural pathway by labeling the input neurons. And we found that the PBPD neurons are anatomically connected to the nodose ganglion, this connection is very important in that this ganglion comprises the vagus nerve, which is 10th cranial nerve that innervates multiple sensory organs, including stomach and intestine. And then we validated the function of the circuit by combining the nerve transection surgery and vibrophotometry recording. And we transected the vagus nerve under the diaphragm, which is called a surgery called subdiaphragmatic vagotomy. And we found that this surgery completely abolishes the responses of the neurons to the gastric distension, whereas the transection of the spinal cord had no effect on their response. So here we could conclude that these neurons receive the mechanosensory signals via the vagus nerve, not the spinal efferents. And finally, we tested the causal function of the neurons on ingestive behaviors like feeding and drinking. And the causality experiment is about the gain of function and loss of function. So in order to do the gain of function study, we artificially activated the neurons by using this excitatory GPCR HM3DQ. And we found that activation of the neurons dramatically suppresses the food intake in food-deprived mice, and also decreased the water intake in water-deprived mice, and both of them are the consequences of the gastric distension. And then we inhibit the neurons to do the loss of function study using inhibitory GPCR, and we found that inhibition of the neurons increased the food intake in the food-deprived mice and water and saline intake in the water-deprived mice, and which means that the activity of the neurons are required for the negative feedback control of ingestion. So in summary, we found a series of circuits from the body to the brain that mediate the mechanosensory signals for controlling ingestion. To the last but not least, I'd like to thanks to my wonderful friends Gyurang and Minyu for leading this project with me, and to my advisor Sungyeon for guiding me in the field, and to all other lab members and collaborators. Thank you.